Hi, my name is Yalin Li, and I'm here to present my research on how seasonal changes affect the reproductive status or nutritional status of Clytia gregaria along the Pacific Northwest. This research was done alongside PhD student Mark Corrales and mentor Dr. Kelly Sutherland. Jellyfish are ubiquitous predators of pelagic communities. These organisms both prey on and compete with other commercially important marine organisms such as fish and invertebrates. Here we have a really quick video showing the live jellyfish with some prey items just to show it kind of like in action. Okay. In the western coast of North America, seasonal changes in wind patterns bring nutrient-rich deep waters to the surface during spring and summer. This fuels phytoplankton growth, which serves as food for plankton. This abundance of planktonic organisms can potentially serve as food for jellyfish. Well-fed jellyfish grow and reproduce much faster, but it is unknown if these plankton blooms could improve the nutritional state of jellyfish. Ishii et al. In 1998 found that starved jellyfish tend to grow much slower in size, but allocate more resource towards gonad development when starved in the laboratory. Thus, comparing the body size to the gonad size of each jellyfish could help us determine the overall nutritional state of their population during different seasons. How we did this was using ImageJ to analyze photos of preserved specimens we collected along the North California current system. Both jellyfish and plankton were collected so that the abundance of plankton can be observed to account for productivity. These were collected using a variety of nets. Mock nets for jellyfish and a 100 micrometer ring net for the abundance of prey, as you can see in these two photos. This diagram here shows how the gonad area and bell diameter were collected for each specimen to obtain information needed for morphological analysis. A straight line tool was used to calculate the diameter, which was then used to calculate the total bell area of the medusa. A freehand polygon, as shown here in the blue, was used to outline each of the gonads to get the total gonad area. A gonadal index was then made by dividing the total gonad area by the bell area. Thus, a higher gonad index indicates that an individual is starved since it has a larger gonads in relation to their body size. The hypothesis that was tested was that when food is scarce, Gregaria will have larger gonads relative to their body size. The hypothesis is supported in our results. From the collected samples of plankton, we found a higher abundance of plankton prey items in the summer compared to that of the winter. In this graph below, you can see that there is a higher gonad area to bell ratio in the jellies collected during the winter compared to that of the summer. You can also see in this graph that there is more variation in the gonadal index in those collected during the winter. In conclusion, the low abundance of prey during the winter might imply that hydro-producing jellyfish are not well fed during the winter season. With this implication, we can then say that a poor nutritional state can be visualized by the use of gonadal index, with starved individuals having larger gonads with respect to bell diameter, due to more energy being expended for gonadal development over somatic growth. In the future, we would like to make a correction factor that converts between the measurements of live and preserved organisms. This is through the preservation method of formalin used, causing a loss of biomass of the preserved organisms, which makes it difficult to relate the morphology of the preserved organisms with that of the live ones. The methods that we'll be using will be based on the correction factor. The methods that we will be using to make a correction factor will be based on research done by Lofontaine and Leggett in 1989, with a graph here showing our predicted results. The green here shows our predicted decrease in overall medusan weight over time, the pink being the diameter and the blue being the gonad area. This will serve as a basis of making a correction factor that translates the current data to data matching that of live organisms. That is all. Thank you for listening.